I'd like to start by introducing today's speaker, Dr. Samuel Lundin. Dr. Lundin is an immunologist and diagnostics innovator and the founder and CEO of Biotome, Perth-based biotech company developing peptide-based precision diagnostics. He was previously a professor at the University of Gothenburg, Sweden, where he led internationally recognized research in Helicobacter pylori immunology and serology. In 2017, Dr. Lundin resigned from his professorship to found Biotome, with the aim of translating academic breakthroughs in precision immunology into scalable, industry-ready diagnostic platforms. He'll talk about one of these projects today in our webinar entitled Development of Helitope, a peptide-based anti-CagA IgG serology ELISA kit. And with that, I'd like to hand the stage over to Dr. Lundin to get us started.
Thank you. Thanks a lot. And thank you everyone for listening in. Good morning or good afternoon or good evening even. I'm in Perth in Western Australia, so it's late evening here. So very excited to talk about this project, the Helitope project. So first of all, I would like to talk about how we view serology and the conventional serology and the limitations thereof. So the normal conventional serology is using whole proteins as targets for the assays. And so here we have a protein, a model protein here with a few epitopes on it. You have different antibodies that bind to different epitopes on this protein. So when then setting up serology test, you have the risk of having cross-reactive epitopes. So some of these epitopes might be present on some other protein from another organism, some other virus, for example, which leads to cross-reactivity in this test towards other organisms, and then therefore a specificity that might be not suboptimal. In addition, the antigens themselves might be poorly defined, we can say, poorly defined in terms of, let's say, as an example, COVID, where you have lots of different variants of the SARS-CoV-2 spike protein. And it's difficult then, of course, to include many of these variants in one test. So if we compare that then to epitope-based or peptide-based, peptide-defined serology, where we focus on single epitopes, single peptides from one epitope in this case, we can then measure antibodies to just these epitopes and be more specific. So this leads then to higher precision of tests. You can limit the cross-reactivity by reducing the number of targets in your assay and be more specific about the targets. You can include strain and variant specificity of this in the test. You can include many different epitopes and many different peptide sequences. If possible, you can also look at disease mechanism targets. So if certain epitopes are specific for a certain disease, maybe an autoimmune disease, maybe a cross-reactivity that actually is associated with a disease function, that can be looked at with this test. You can also, it's a more cost-effective and scalable synthesis of these targets because the peptide synthesis is relatively cost-effective. And you can even patent the use of these epitopes even for well-known antigens. So normally we would say that protein-based serology is a bit like having a high-resolution camera with a blurring filter in front of it. The immune system is highly accurate and diverse and specific, but you're blurring the filter. You don't get the actually full use of the immune system. While with selected epitopes, you get a more clearer picture of the thing you want to look at. So you use the full specificity and diversity of the immune system. So this test then, the Helitope test, is a CagA antibody test. We measure antibodies to the CagA protein. And just very quickly, it's a relevant marker because this is a bacterial oncoprotein. It's associated with virulence of the infection, but also gastric cancer risk. So CagA, so H. pylori itself is present in half the world population. And some of the H. pylori strains have the CagA protein. And a subset of these people with the CagA infection, they develop gastric cancer. So it's a highly relevant protein to make a serology test against. But there are existing challenges for this protein. It's a highly variable antigen. It's equally variable to many of the viruses even. So it's very variable. And the tests that are out there today have been very inconsistent in terms of specificity. So there's room for a better test against this protein. So then we set out to develop such a test, and we are using this sort of 6-point pathway or pipeline to create serology tests. And the first 3 are related to peptide arrays, so we'll go through those, and then we'll go through the rest at the end as well. So first of all then, we are using epitope mapping using peptide microarrays, and of course that's why we're having this webinar with Pepperprint, our long-standing partner in this area. So the first of all, we use, depending on what the target is, we use different numbers of peptides as a first step, as a screening step. In this case, the CagA case, it's just one protein, so we can aim for around 1,000 or 1,100 peptides, 12 mer peptides with an 11 amino acid sequence overlap between each of them. And that covers the entirety of the protein. We put these peptides on random positions on the array in order to get as good data as possible. But if we're doing this experiment with a larger protein or a proteome, we can get up to 100,000 of peptides in one go in this screening stage. So now we test, in this stage, we test usually pooled serum samples to be more cost effective in terms of testing these large numbers of peptides. And of course, it's really important, as in any diagnostic development, the samples you have. So in this particular case, then for CagA, it's slightly complex. So the gold standard, the only way of knowing if you have CagA or not, is really to do DNA sequencing of H. pylori isolate from gastric biopsies. So it's a bit of an endeavor to actually get hold of these types of samples. You need to select biopsies from patients. You need to culture the bacteria from the biopsies and then sequence them. But we've did that in this case. So we use then pooled samples, as I said, usually between 5 or 10 samples in each sample pool. And the purpose of this first step, the screening step, is then to identify all the antibody epitopes used by these samples. Not really too much concerned about which ones are useful for diagnostic or not, but just to identify all of them. And the simple way we call an epitope is really to see if there's a consistent increase in signal in consecutive peptides along these overlapping peptide sequences. This is just an example of how things can look. We use different types of arrays. So they could be the 5-sector medium density arrays with 1,400 peptides, or more lately the 1-sector, the high density arrays with many more peptides on them. But typically we test a couple of different antibodies at the same time. Usually it's IgG and IgA using different secondary reagents. So you then get different colors of these little peptide spots on the arrays. But the outcome really of this test then, of this screening phase, is a map of the epitopes across the sequence of the protein. So here we have the CagA protein, which has the 1100 amino acids. It got several different domains, structural domains, and we can see on the y-axis here the signal intensity on the array. So basically what we can see from this case then, comparing, well, looking at the H. pylori infected serum pools and also the uninfected serum pool, is that there are many linear CagA epitopes. Obviously, we count roughly 18 of them with various intensity. Interestingly, in this case, most of them are in this C-terminal region, the unstructured, non-structured C-terminal region. And the strongest epitopes, also interestingly in this case, are different functional sequence motifs. So the protein has certain motifs with certain functions that when this protein goes into epithelial cells of the gastric mucosa. So these motifs that have functions in the cells and affect the cell signaling, they are also epitopes in this case. All right, so that's the screening phase. We've identified the epitopes. We know where they are, how big they are, and what sequences they have. So then we move to second or third phase then of this pipeline is to identify the most discriminatory peptides. And then we again do another round of arrays with now fewer peptides. We select peptides from each of the epitopes, several from each. If known, if we have the capacity, we include known sequence variants as well of these epitopes. This time we identify or we test individual serum samples, not pooled, to identify the diagnostic capacity. And we measure the AUC, area under the curve, in the difference of signal between the cases and the control, or the positives and the negatives. For patenting purposes, we might also include here, either in this round or in a following subsequent third round, synthetic variants of, oops, sorry, now I lost my thing. Here we go. Synthetic variants of the epitopes in order to, it's because the epitopes or the peptides we use are roughly 12 or maybe 15 amino acid long. In order for patenting to work, we will need to shorten this to the most shortened, the shortest possible motif that we can protect then in a patent. So to do that, we do systematic variation of these sequences by either alanine scanning or substitution of amino acids on these sequences. And then that leads us then to the sort of final result in terms of epitope mapping. And here we see the AUC plotted. So each dot in this diagram is one peptide from one epitope. So all of these are epitopes with antibody responses to them. And you see the majority of these epitopes fall in the AUC between 0.4 and 0.6, which means they're actually useless for diagnostics. And this is really the reason why, at least in this case, CagA case, that whole protein serology is not so successful for CagA. So if we would use all these epitopes, which you would if you have a whole protein on your assay, you'll end up with a quite fuzzy image, which is not very discriminatory between cases and controls, or infected and non-infected. But if we can select just the best epitopes, that's when you get a more effective serology test. So the key here then is that only a minority of the epitopes are in fact discriminatory, and that in itself is key for patentability. So because we can show in our assays that just a few of these epitopes are useful, that means that fact is patentable. So we can then patent those ones, which we have done. All right, so now we've covered the epitope mapping part of this workflow, and then number 4, we I won't dwell much on at all, but that's in order to have a functional test, we need, of course, positive controls or calibrators in our tests. So in our case, we then generate monoclonal antibodies to these selected peptides that we've selected, and those monoclonals can then be used later on as positive controls or calibrators. But we'll move on to the last two steps here, which is the peptide modification and ELISA optimization. And there are then important challenges when you're translating peptides from these hits from the arrays. We know there are hits in the arrays. We know that samples react to them. It's very consistent. They've been positive in several tests. But then there's a challenge of, okay, how to put this on an actual assay platform and get measurable results from using peptides. And mainly revolves these difficulties around surface immobilization. You can use a few different approaches to put peptides onto a surface. So you can use passive adsorption, like with protein. You just chuck it into a high bind plate, for example, and they will to some extent sometimes adhere to the plate. A better approach could be to use chemical coupling, EDC and HS coupling, in case your peptide has lysine residues, for example. But still, it's not really optimal. You still get random peptide orientation on this surface. There's also problems with manufacturing complexity and variability with these approaches. Another very common way, of course, is to have biotin-coupled peptides. So here you have a biotin moiety on one end of the peptide, and that can, of course, stick to streptavidin, for example. However, it's not ideal for proper serology tests, proper in vitro diagnostics, because there is background that's introduced by this problem because of biotin that could be present in serum samples and so on. So it's not ideal, and it also has manufacturing complexities included in that approach. And just to show the background aspect of this then, so here we've tested, this is just an example of a few different serum samples, one on each bar. The approach or the model here is a polio peptide with a biotin on it in streptavidin plates or nutravidin plates. And you see the black bars is the background. So different serum samples have variable background in this setting. Although, of course, there is a signal, there is a positive signal to the polio peptide, you also get background that varies between samples. Another approach that could be used is maleimide or cysteine-coupled peptides. So here's a polio peptide with a cysteine end. But that, in our hands at least, create even worse background. So it's not an ideal way of doing things. So in order to get over this difficulty, we then decided to develop another way of doing it. So we came up after a couple of 2 or 3 years of actually innovation and testing with this approach called PepLock, or we call it PepLock. So this is a peptide-based one-step immobilization chemistry. So it's a moiety at the or a modification or a series of modification at the end of one side of the peptide. And this is now being filed for patents. So this enables then targeted, very rapid and very stable display on an assay surface. So what we now then end up with is we can put the epitopes on the bottom of the assay surface. We can then add serum IgG, they bind to the epitope, and we can detect using normal ELISA method, normal indirect ELISA. And just to show the results of this then, if you have a PepLock modified or versus unmodified peptide, these are model peptides, you can see a really, really big difference in signal strength to these peptides. So it's an effective approach, and we're actually surprised by how well it works. And background-wise, it's also quite favorable. We don't see much background with this approach at all. So going back then to the CagA story, CagA approach, we have selected peptides using the array tests, and now next step is then to test these or validate these and select ultimately the peptides used in the assay, in the ELISA. So here we have tested a top list of the best peptides, including no peptide at all. We're testing here the blue are CagA positive samples, so samples from known verified CagA positive infection using sequencing, and the red is the CagA negatives. So there's a number of peptides that work pretty well, and the question is what to choose in your ultimate test then. And then we ended up with selecting 4 of these based on partly on the signal to noise ratio that we found with these ones, but also the sequence coverage. So as I said, CagA protein is a highly variable protein, and H. pylori and CagA has a geographical aspect to it. So different strains from different geographies have different inherent sequences, because H. pylori followed humans probably already from out of Africa and then spread around the world through history. And you more or less inherit your H. pylori strain from your, probably most from your mother, but at least from your family. So here we have the 4 different peptides we selected, and the coverage, the sequence coverage of these individual peptides is indicated by the blue scale here. So not only did we have to test or choose peptides based on the signal in the assay, but also something that most likely would cover CagA sequences from around the world. That remains to be seen, but that's the idea behind the selection. So now we've tested a couple of different cohorts from a couple of different areas. We have tested a Central American cohort and a European cohort, but we need to keep testing other cohorts as well to see how well this approach works. But that's the basis from the selection. And then finally, then we've ended up with a manufactured test from this. So we've patented these markers, as we said. It's now granted in 20 different countries. We've developed the assay at Biotome, and we use this 4-peptide mix. So there's one well with 4 peptides. Of course, principally, you could have several different wells and have a multiplex test, either in ELISA or you can put it on a Luminex background. But in this case, we've selected one well with 4 peptides in. We also use a dual test and background well setup. So we have 2 wells for each sample, one with a background, one with a test, in order to get a more higher accuracy and more specificity of the test. So we took the assay so far, but then there was we sent this to our partner Biosenses in Adelaide in Australia, and they did further optimization and manufacture, and they're now also marketing this test very soon. They've proven the stability. It will be launched as a research use only kit very shortly. And the results that we have from these from this kit, then from the cohorts we have tested so far. So this Central American cohort, which is the one, it's from Nicaragua. It's the one where we have sequencing information from the cultured H. pylori from these patients. So it's a very, very well-characterized cohort. And we're testing those with a known CagA infection. They are positive to 96%. So we have a 96% sensitivity in this cohort, while the CagA negatives, which still have an H. pylori infection, they are negative, all of them in this case. That's a pretty small sample cohort though. So looking at specificity, we have also, as I said, tested a European, in this case, a Swedish cohort, which is larger. Now, this is then not ideal in terms of sequencing. There is no sequencing information. There's no culture from this cohort. There is a H. pylori serology data associated with it. So we know who had H. pylori infection and who did not. So, and we also know that roughly 50 to 75% of H. pylori infected are expected to have CagA infection. So we get a 69% result or positive result in this cohort. But the important, I think, number is this one. So those without a known H. pylori infection, they are positive to only 7%. So the specificity based on this data is at least 93%. So these positives are either false positives or they had been exposed to CagA H. pylori in the past. But at least we can say the specificity is 93%. So finally, the final data point, if you compare this then to an existing CagA assay, sorry, which is the Helicoblot Western blot assay, from the same data from the same cohort, they found 94% positive of the H. pylori infected and 41% of the non-infected. And these numbers are unrealistically high. It's unexpected to get 94% CagA infection in this European setting. And it's unlikely 41% actually should be positive to CagA. So it's most likely these are false positives. So based on the evidence so far, we can say that the specificity we get is high, and it's most likely higher than this existing test. So that leads us to the summary. So I would summarize these data, these results, and our experience as saying that peptide arrays surely can enable development of high specificity serology assays. Also that, of course, there can be different approaches, but you can easily mix several peptides into one test, into one well. But you could also theoretically do multiplex versions of it as well with orthogonal testing of different peptides or different groups of peptides. There are surface immobilization challenges of peptides, but they can be solved using our PepLock approach. And finally, that we can, I think this demonstrates that there's a possibility of an end-to-end translation of manufacturable assays using the approach that we have taken. And then finally, thank you to our contributors. We have a large range of academic collaborators in University of Gothenburg, in Nicaragua, in Toronto, Canada, in Houston, and here in Western Australia, and also in a Swedish regional hospital. Pepperprint has been very helpful, of course, providing arrays to these experiments for a long time. So it's always been very, very helpful, very high quality arrays that's been delivered. We're very happy with those. Finally, Biosense then in Adelaide, our partner who's doing the manufacture of the test and the marketing of it. And of course, thank you to the amazing Biotome team that's been either today or previously been in part of this project. And you can see my email address at the bottom there if you have any questions after this talk. So thank you. Thanks for listening, and happy to take questions if there are any.